I work a lot on single-cell sequencing, so that's the method that I pioneered a lot when I was a postdoc at Cold Spring Harbor, and also my lab now at MD Anderson focuses a lot on single-cell sequencing. That's one method in which you can sample single cells and get genomic information and infer tumor evolution, and also to help guide things like therapy. Other methods include deep sequencing, so you can sequence at very high depths. You can detect rare mutations that are present in the tumor mass. And another method that's been quite popular is spatial sequencing, where you take different regions within a tumor and you perform exome sequencing, and you look at genetic differences that occurred as those tumor cells migrated and evolved throughout the tumor. And that can also guide things like therapy. So those are kind of the big sequencing methods that people use right now to study clonal diversity and substructure. So our data so far, what I presented showed that there's a lot of very rare mutations that occur in the tumor mass. And we show that by single-cell sequencing, but also by deeply sequencing in the tumor. And even in cancers like estrogen receptor positive breast cancer, where there aren't a lot of mutations that occur, you can still find these rare mutations that are 1% or less of the tumor mass. And because they exist in the tumor at a low frequency, when you challenge with therapy, you eradicate most of the tumor, but it's very likely that some of those resistance mutations are present and they're going to grow back and expand afterwards, because in most patients right now, after chemotherapy for 2 years or so, the tumor can regress and the mass can shrink considerably in size, but it always grows back and becomes resistant. So our data suggests that a lot of these resistant mutations are pre-existing in the tumor mass and can probably account for a lot of the resistance that you see in breast cancer. So for single-cell sequencing, the big challenges are being able to do it in a cost-efficient manner. So getting the cost down so that you can easily sequence 100 or 1,000 cells from a patient's tumor and get a good map of its diversity. Also, the technical errors are still a big problem right now. So there are a lot of false positive and false negative errors, and we have to do more technology development to get really good, robust, accurate information if we want to translate it into the clinic and actually use this for patients. So those are kind of the big barriers right now. I've been working at Cold Spring Harbor, where we've collaborated with Annelies Borensendale for quite some time now, over the past decade actually. And I know that for breast cancer research, it's really, especially Norway and Oslo are one of the top leading research institutes, and it's just phenomenal how her group and others have been able to collect a lot of samples with all the clinical information and have these large cohorts of patients, and also how willing the people in Norway are to provide their samples for research and for discovering biomarkers. And so I know that it's a very good medical record system where you have follow-up information and the patients are willing to come back, and also you can track them over long periods of time. And in the US, you might have that within one hospital, but you don't have that usually statewide or nationwide. So you're trying to pull together a lot of information that can be very disorganized. So I know for breast cancer research, it's really a great place to be, and there have been some pretty high impact papers that have come out of Norway, especially one that I was mentioning in my talk, which was on the original gene expression patterns in breast cancer patients. And it's one of the most highly cited papers in scientific research.